Thank you very much for the kind introduction. That sounds like a really bloated academic CV. Much to that. But so I'm the chair of the biological psychology and neuroeconomics here at Technical University of Berlin. And we're trying to understand psychology using biological approaches, or trying to understand how humans behave and experience, and look specifically at the biological basis of that. And one of the foci that we have basically is electroencephalography, measuring the human brain activity, electrical activity of the human brain. And we're trying to understand how natural cognition and multisensory information processing actually in the human brain unfolds using these kind of approaches. And if you look at, okay. Just a second. So let's try this. So if you look at how we usually investigate human brain activity in humans, basically, obviously human brain activity, then we force participants to lie supine in a scanner that's a functional magnetic resonance imaging on the left side. On the right side, you see a typical EEG setup where people are seated in front of a monitor. They're not allowed to move at all in neither of the traditional brain imaging modalities, because movement leads to a movement of the source of the activity that we're trying to measure, that is an artifact, and that makes it pretty much impossible to understand what's going on. So if you look at that, we record millions of bits of information about brain activity, and we try to compare that to one bit of information regarding the behavior. That is usually a button press at the end of the trial. And this is not what happens in our natural environments during daily life activities. So we actively move through the environment, interact with our environment, and use our full physical structure to reach cognitive goals from an embodiment perspective. This is a very artificial setup that we use to understand the human brain. If you look at animal studies, however, the last decade and last 2 decades basically have shown that the animal brain dynamics change drastically the moment that animals start behaving. If you look at the fruit fly here, Drosophila, the moment this fruit fly basically is glued to a stick, and Beard and Dickinson here in that experiment recorded single neurons in the fruit fly mushroom body. That's like a brain area that mediates between visual input and motor output. It's not directly related to visual processing or motor control, but something intermediate. And if you look at the single neuron activity when these animals are resting, some of these cells are entirely quiescent. There's no activity going on. The moment that the fruit fly is airborne, the dynamics drastically change and these single neurons become really active. And this happens in a number of species, that the brain dynamic state changes dependent on the behavioral state. And what we and Dickinson coin here is that the neuroarchitecture, so the hardware, the connectivity between different brain areas and neurons obviously does not change, but the connectivity and the information transfer between different regions drastically changes dependent on the behavioral state. So how can we then investigate that in human brains? If you go back to the traditional fMRI or EEG study, that obviously seems pretty difficult. But the last years have shown a rise in technology that makes this possible. And this is one of the last examples 2 years ago by Barbara Hindel's group in Würzburg, basically showing that if participants walk as compared to standing, the brain dynamics also drastically change. What you see here is a spectrum. It's alpha activity around 10 Hertz in black, where people are standing still in a light hall. It's a standard, was like a gym, basically, where they were standing. They were slow walking in green or normal walking. What you can see is that there's a massive alpha reduction when participants start walking. And even if you switch off the light, this is still the case. So there is a strong modulation dependent on visual input to the visual cortex, but simply walking changes the brain activity of participants drastically. So that's a summary on the left-hand side. In red, you can see the dark condition, and green, the light condition. still means just standing. SW is slow walking and then normal walking. And there's a drastic change that you can see, independent of the light that enters the system. So in general, again, coming back to the technology, how do we investigate then human brain dynamics in actively behaving participants? And this is basically one example from our lab. So in the background, you see our Berlin Mobile Brain Body Imaging Lab. That's a dedicated lab space. This lab here has 150 square meters that we can basically use to have people walk around in virtual reality, any kind of virtual worlds that you can create. But that still provides you with control of what participants see and when they see it. So there is a higher ecological validity. People can move, and we still have some kind of control over what they perceive in these environments. And what we do here is we use mobile electroencephalography. You could also use functional near-infrared spectroscopy. These are the only two systems at the moment that allow mobile brain activity recordings. And then we couple that with motion capture because we want to understand how active behavior influences human brain dynamics and how human brain dynamics make use of active behavior to reach, for example, cognitive goals. Then, in this case, we use head-mounted virtual reality, but we also do that outside the lab in the real world. And usually what you want to know is some kind of context information. So what do people attend to? What do they see? So you have to synchronize eye tracking with the other modalities that you record. And then you can use whatever you want. Electrocardiography or electrodermal activity to get for the peripheral physiological measures, good indicators for stress, for example. If you think about neurourbanistic research, when people walk through real cities to understand how urban stress impacts human brain activity. And then obviously this is like a tactical challenge. And I think I would say like 1/3 of the time that we spend in our lab is trying to get over technical issues. So there are like different data streams coming in with different sample rates. Because everything has to be wireless, this is non-trivial. Rendering of virtual reality depends on the complexity of the rendering and might delay different samples. Things like that can be overcome, and LSL here is one example of how this is done. This is the lab streaming layer architecture that basically provides one mother clock and streams everything that's up in the air. Any kind of data stream that has Bluetooth or wireless connection can be streamed and synchronized using that kind of software. And then obviously also the analysis is quite complex. And I'm not going to dive into the details here, but what you see here in this simulation is like two simulated sources in a human realistic head model. This is a brain, basically. This is two artificial sources that we modeled in the brain. And if you have a transfer function of the brain through the skins and the skull to the electrodes on the surface, this is what you would see. And considering that there are two alpha frequency sources here, this is like the right parietal cortex oscillating a little bit slower than the left motor cortex here. But if you both back project to the surface where we would record the activity, what you can see is that there is no way to differentiate between the two sources being active at the same time. And obviously, there are not only two sources being active, there are several sources, and there are also extra brain sources, muscle, facial muscles, mechanical artifacts, and all the other things that will happen when you start recording a mobile participant. So we need analytical approaches, data-driven approaches that allow us to differentiate between these data streams. And I'm not going to dive into that today, but I would like to give you an example of what that looks like and why multisensory information processing is a more natural way of reaching your cognitive goal than lying in a scanner or sitting in a dark room. This is an embodied heading computation experiment. So we put like all the things that I just talked about on our participants, and we have them rotate on the spot. I really hate that when I have to do that sitting in the audience, but I would like to ask you to stand up just for a second. And I just want you to have a very short rotation on the spot until I say stop. Just close your eyes. And when I say start, you start rotating. When I say stop, you stop rotating. And after that, you still close your eyes and you rotate back and point to me still being here. Okay? So close your eyes. Start. Stop. Now rotate back without opening your eyes and point at me. Okay, now you can open your eyes. And you see you did a pretty amazing job without any visual information. I mean, obviously I'm talking, there's like a sound source that helps you, but I can do that even without sound. I can close my eyes, I can rotate around, and I still know that this is more or less the initial heading that I have. And this is because we have multi-sensory information about that rotation and space. Our inner ear, the vestibular system, gives you information about acceleration, 3 dimensions. We have proprioceptive feedback about the muscles and the relative position of the head to the trunk, to the shoulders, everything. Plus acoustic input, plus visual input. This is the natural multi-sensory processing that we would use when we try to estimate our heading. So this is what people did in this experiment. So what you see is they're standing on the spot, then they see an orienting beacon, that is their initial heading. This is then replaced by a sphere. I'm sorry, something is not working here. So that is replaced by a sphere, and this sphere then starts rotating with different velocity profiles, ends unpredictably on each file at different eccentricities to the left or to the right, and then changes its color, and that's the imperative stimulus when participants have to rotate back and indicate their original heading. And this is basically then what we use to compute the heading errors, so under an overestimation of how good they actually are. We cluster that for statistical power reasons for computing then the difference between the conditions. And in general, there's not much of a difference between left and right. So everything that I show you here is aggregated over left and right rotations. And we're looking only at the outward rotation. Importantly, what we do here is we always compare the mobile condition with a traditional desktop condition. And this is what is usually done in the scanner or in an EEG lab. People are standing and using some kind of input device to control the rotation because you're not allowed to move. And this is what we do usually to have a comparison of what differs now. If you do the same task, it's the exact same visual input that people see, but they actively rotate as compared to using a joystick to rotate. Excuse me. So what you see here is the absolute heading error as a function of eccentricity of the outward rotation. So people rotate out 30, 40, 50, up to 150 degrees. And this is the absolute heading error. And in blue, what you can see here is the absolute heading error for the stationary 2D with the joystick control. What you can see is like a more or less monotonic increase of the absolute heading error with increasing eccentricity. This is what we usually see in these desktop experiments. But from what I just saw, you doing this task, you were pretty much on the point, right? If you compare that to the mobile condition, what you can see here is a drastic change. People are much, much better. There's a significant difference in the accuracy between moving and joystick. There is a significant main effect of the eccentricity. There's still more or higher errors with higher eccentricities, but what you also see is that up to 90 or 100 degrees, there's no increase in the heading error. And this is as long as you don't rotate out of your cardinal body axis, you basically can use a very accurate representation of your heading. The moment that you rotate beyond your 90 degree or this 180 degree plane, you start obviously switching to a different spatial representation to solve this task. This is quite interesting, but what's more interesting then obviously is the brain dynamics that happen during this kind of task. And I'm already at 50 minutes, so I run through that.
You still have to.
Pretty quickly. I'm just trying the idea that we had here was if it is vestibular information that you use in proprioception, then there should be some kind of velocity or acceleration dependent signal that is visible in the brain. So what we did, we basically used the motion capture information and chunked the outward rotation according to the velocity. And then we sorted from the 10% lowest velocity trials to the 10% highest velocity trial, all within one rotation, so different velocities. And we computed the power of different frequency bands during these outward rotation and then sorted them also according to the velocity that was going on. And what you can see here then is like that in the physical rotation condition from 3 to 30 hertz. This is the sorted power of all these frequencies. Here is an example for the theta range from 3 to 5 hertz, with the 10 lowest up to the 10 highest velocities. And if there is a striping pattern with different colors, then obviously this would indicate that there's a power modulation dependent on the velocity of this rotation. And I'm just running through that. It's tons of statistics here using dissimilarity matrices, computing the Mahalanobis distance between all these different frequencies in all these different bands and dependent on the velocity. And then you look where the differences are. And in this, you see still the frequencies sorted according to velocity. And in the upper left triangle, we compare only different frequencies depending on different velocities for the mobile condition. The lower left square compares both the MOBI and the joystick condition, and then the lower right here would only be the joystick condition. The diagonal then obviously is a mirror image of what you computed on the left side. Then we run some statistics and see what happens here. And this is original data. This is data from activity that most likely stems from the sternocleidomastoid muscle. This is like the sternocleidomastoid. That's a muscle that attaches to your mastoid process, goes down to the sternum. And when you contract that, you basically rotate your head down and to the left or to the right. So it is a muscle involved in head rotation. And what you see here is that there's tons of differences in this muscle activity in the physical rotation condition for different frequency bands that differs maximum between the joystick condition and the MOBI condition, but there's also differences depending on the velocity in the different frequency bands for the mobile condition. And this makes a lot of sense, but if you look at the stationary condition, there's absolutely no modulation dependent on velocity. Makes sense because in the physical rotation, you rotate your head. Different velocities should have an impact on the brain or the muscle activity. But when you use a joystick, there is no movement of the head. Thus, the muscle should not be active. And this was like a proof of principle for us that this works. And just one example here is the visual cortex. As another example, what you see is there's not much going on dependent on the velocity in the visual cortex. And now the pattern more or less inverts. For the mobile condition, your visual cortex is more or less inactive. But for the joystick condition, there is massive differences depending on the velocity of the sphere that you see. Again, if you come from a predictive processing perspective, you know what you are going to perceive sensorial-wise when you rotate actively. And as long as everything that you predict during that rotation comes in sensorially, there is no activity. But in the joystick condition, you basically just have a visual change, and that depends on the velocity. So that was an example, and the general idea here is that if we start moving, the simple movement-related sensory feedback will be processed by the brain, and thus brain activity during active behavior will drastically change from stationary setups. That seems to be very plausible and simple. So we will have to expect different brain dynamic patterns when we go out and have participants actively behave. If you then go into the real environment, like we also do, then you lose control over external factors like cars honking at you, or this is like a study that we run in Berlin. Sometimes people look at you, not very often in Berlin. Other cities might be different. Here, nobody really cares, but it basically shows that we are able to leave the lab and start getting ecologically valid data during active behavior in the real world. And that will change what we see because behavioral states of participants influence the brain dynamic states. We will see potentially a reflection of the early, very classical evoked activity pattern because the neural wiring still is the same, but everything that happens later and will be feedback by sensory information that comes from active behavior will change. And I think in combination with other assessment methods, this is a really good method to now go beyond the lab and get a better understanding of natural cognition, natural affective processes when we act in our environment. Sorry for the slide overlay. Thank you for everybody working on that. And